Hi, my name is Mike McDonald from the University of Tennessee, and this is the APS Convention 2013 in Washington, D.C. We are studying the executive dysfunction in a mouse model of Parkinson's disease. And typically people think of Parkinson's disease as a motor disorder, and it is, but half the patients also have cognitive dysfunction, characterized by deficits in working memory, impulsive behavior, planning, short-term working memory, and so on. And so we first trained our mice on a 3-hole serial reaction time task, which is a test of sustained attention and impulsive behavior. And to do this, on each trial, a house light lit up above, which was their signal to get ready. And then after a randomized delay, one of 3 lights flashed on very briefly, also randomized. And the hungry mice had to go poke their nose in the hole in order to get a food reinforcer. After we trained them to do this, we injected half the mice with MPTP, which is a neurotoxin that destroys part of the brain that is damaged in Parkinson's disease. And after these lesions, the mice were still able to do the task pretty well. And so we challenged them with more difficult conditions. So first, we reduced the cue duration down to as low as 1/8 of a second. And when we did this, the MPTP lesion mice had slower reaction times at the shorter cue durations. We also increased the duration of the get ready period. And when we did this, the lesion mice were still able to do it, except under the very long cue durations when impulse control broke down and they started responding prematurely. And so then we examined their brains to verify the lesion, and we found that in fact they had substantial destruction of the substantia nigra and loss of dopamine in the striatum. And we correlated all the behaviors with the neurochemical measures in the striatum and prefrontal cortex, and we found out that serotonin turnover in the prefrontal cortex was strongly correlated with impulsive behavior and slower reaction times only in the MPTP treated mice and not in the saline controls. And in fact, none of the other neurochemical measures was correlated with any of the behaviors in either group. And so it's fairly well known that impulsive behavior is mediated by serotonin turnover in the prefrontal cortex, but it was kind of surprising to see that reaction times were also associated with prefrontal serotonin turnover. And so taken together, these results show that we can use this serial reaction time task to assess and study the executive dysfunction associated with Parkinson's disease, and that we can also look at the neurochemical measures to try to understand the underlying mechanism of these behaviors.